Yes. I don't know what was the problem. I have no idea, Patrick. I'm sorry. And then definitely it was not you. Okay, so tissues, there are 4 different types of tissues. You already know that. We talked about it quickly. Epithelial tissue, muscular tissue, nervous tissue, and connective tissue. Anything, this is the definition that my physiology, my histology teacher gave me. Anything that is not epithelial, muscular, or nervous tissue is connective tissue. During the exam, if you cannot recognize a tissue, write down connective tissue for the lab exam, right? Write down just connective tissue. Because anything else that is not epithelial, muscular, nervous tissue, it is connective tissue. So let's go with epithelial tissue. Epithelial tissue, they can be simple. What I mean by simple, of course, there are 3 types. Squamous, it means flat, right? They're squamous, they're flat, they are cuboidal, like a cube, and they are columnar, like a column. Okay, so there are 3 basic shapes of epithelial tissue. Now, in our body, they are stacked in a way. If there are a single layer of them, just one layer of them, whatever it is, then it's called simple, cuboidal, columnar, simple squamous. And you do have all 3 of them in your slide collection. Simple. You just, in the lab practical exam, you cannot say it's a squamous epithelial tissue. No, it is simple squamous or stratified. If it is more than 1 layer, it is called stratified. So simple squamous epithelial tissue, they are found in your mouth, lung, coelom, which you don't know what that is now, but you will enter your blood vessels and so on and so forth. Okay. Cuboidal epithelial tissue, they are found in your slide of kidney. We do not have slide of salivary gland, and we have slide of thyroid gland. We do have them on your slide collection, on your slide microscopic slides. When I say slide, microscopic slides. Okay, and then columnar epithelial tissue, they are found in your digestive tract. Okay, we do have slide of it. We do have model of that one, and many glands, goblet cells, and then stratified epithelial tissue, which is found in your skin, and you will see it in a few places, when they are stacked on top of each other, when you have more than 1 layer, then when they are stacked on top of each other, then it's called stratified. So on this case, how do you know, is it columnar or is it cuboidal? Well, if the bottom layer and the bottom right here, it would be connective tissue or other parts of the body, and right here usually is an empty space, your gland, your blood vessels, what have you. Then on top, whatever is on top, then it is that type of epithelial tissue. So what would you call this one on top of each other? Cuboidal on the bottom and columnar on top. You call it stratified columnar epithelial tissue. I hope I'm making some sense. We do have some slides, and hopefully those slides make some sense. Okay, let's go over and look at what else. Here we go. Is this diagram has, for example, pseudostratified ciliated columnar epithelial tissue, which is found in your trachea. Okay, it is found in your trachea. They are not showing it here. It is ciliated, okay, and it looks like stratified, but it is not stratified. It is 1 layer. That's what they call it, ciliated pseudo. Pseudo, it means false, right? On your prefix, suffix, pseudo means false. You think, when you look at it on a microscope, you think it's more than 1 layer, but it's not. It's actually 1 layer. And then you have pseudostratified columnar epithelial tissue in your trachea. You have stratified squamous epithelial tissue. You do have a slide of that, microscopic slides. You should start looking at them today. Or you should have started looking at them, I'm sorry, last Monday, on Martin Luther King Day. You should have took some break, you know. But remember, a student is what? 365. You only get 2-3 days off. Martin Luther King Day is one of them. Thanksgiving is another one of them. And Christmas is another one. I told you my botany teacher, he changed it every time he gave that sermon. Next time he said, oh, it's Labor Day, it's Independence Day, I don't know, he said St. Patrick's Day. He always made it different, but anyway. So you're supposed to study 365. if you're planning to go to medical school, pharmacy school, dental school. Anyhow, so right here is stratified squamous epithelial tissue. It is found in your esophagus. You do have slide of esophagus and trachea next to each other. Okay, it's more than one layer, and all of it is squamous epithelial tissue. Then in your lungs, you have simple squamous epithelial tissue. You do have slide of it again in your lung. And then your kidney, you have simple cuboidal, you have slide of it. And your intestine, simple columnar, you have slide of it. And then of course in your bladder, it says stratified columnar, it's not. See on the bottom, it's cuboidal, you guys see that? And on top is what? Columnar, so you call it stratified columnar, but he's wrong. It's not your bladder, maybe in monkey it is stratified columnar, but in us it is transitional. So, and I gave it to you guys. It's in your handout. The handout that I gave you, slide description, it is transitional epithelial tissue. It changes its shape. Those epithelial tissue change their shape, like accordion. You know what I'm talking about? You know what accordion is, an instrument to play. So it changes its shape. Yes, Farha.
If cuboidal was on top, it would be stratified cuboidal, right?
That's right. Whatever is on top. Okay, here is the real this was a cartoon, but this is a real picture of simple cuboidal epithelial tissue. It's one layer. Why is it one layer? Because this is one follicle, and this is another follicle, and they lie next to each other. You might think it's more than one layer, but it's not.
How do you know it's cuboidal?
Look at the nucleus. It's
okay.
Nice. It's cuboidal. Very good. I don't want to waste too much time of the video, then it's hard to. 100,000 people were cheering the football team of Tennessee, and I was in the lab, the biology lab building was next to the stadium, and they were cheering Tennessee anytime they made a touchdown or anything. And here I was looking at the slides on weekends, on Saturdays. Usually college games are on Saturdays.
Oh, Saturday.
College, Saturday, pro, Sunday. Pro nowadays is every day. Thursday night. During my time, when I used to follow and watch, I had more time. Students have more time than ordinary people, I guess. It was on Saturday. So, and I was in the lab looking at the slides. You have to look at them. And I'm sure I will help you out, I'll hold your hand, but you have to keep looking at them. Once or twice, it will not cut it. You have to keep looking at them, day in, day out, every day. Okay, here is a stratified, it's more than one layer of columnar epithelial tissue. The top layer looks like column, on the bottom it could be cuboidal, so on. Muscular tissue, you guys did a good job on it. I think we covered it up pretty good, but skeletal muscle, striated and voluntarily, okay? It is right here, a few characteristics of skeletal muscle are here. Skeletal muscle are striated. You cannot see it on the video, but you can see the line. The nucleus is on the periphery. More than one nuclei, one cell, more than one nucleus. In Bio 1, you learn most cells have one nucleus. Here, more than one nucleus. Okay, you have many nuclei. Then it's striated, it's voluntarily. You do have control over it, and so on and so forth. Smooth muscle, we have a model of it. Again, you cannot see on the video, you cannot see this, does not have striation, but right here, if you look at it, it's smooth, there is no striation. And later on, when we get to skeletal muscle, I'll let you know why striation. Okay, why you can see the striation on the microscope. We do have skeletal muscle on the microscope. We have smooth muscle on the microscope. You all know what I'm talking about when I say microscope. And then, of course, one nucleus, one cell. And the nucleus is right in the middle of the cell. Cardiac, striation. Okay, cardiac, there is striation. And of course, non-striated, non-voluntarily. Usually smooth muscle are in your tubes of the body. The tubes, okay, whatever tube you have. And then cardiac muscle only found in the heart, nowhere else in the body. Striated, they have one nucleus or two nuclei per cell.
What is striated? One
or two. We'll talk about it when we get to skeletal muscle. Okay. It's actin and myosin. I don't know you studied it or not. It's actually the myosin you can see in the microscope. Okay. Then the nucleus, one or two, is branched. Some of the cells are branched. I talked about this. And then, of course, cardiac muscle pumps the blood to the rest of the body. Okay, cardiac muscle. Striated only in heart. I hope I said it all. Here are striation. You can see that, and we'll talk about it, actin and myosin in a minute, but not in a minute, not today. But these are the lines you see, the nuclei. This is a real picture, this is not a cartoon, are in the periphery of the cell, and so on and so forth. Okay, the next tissue is nervous tissue. I left the connective tissue, I guess, for the last. Here is a neuron, and the neuron has dendrites. These are the dendrites, and this is the cell body, and this is the axon, and at the end of the axon you have the axon terminal, and at the end of the axon terminal you have axon knob. Okay, so this is a model of a neuron. This one, I don't like it. I like it, but what they've done, I think, is a mistake to cut the cell body, they call this cell body, from the axon. And students think that they're two different things. No, they're connected. You might think that they're cut, but no, they're connected, just like that model. You know what I'm talking about? I want to make you some sense. Just like this model, it is connected, but in this model, they disconnected. How about that? Cell body, axon, these are dendrites, these are axon terminal. At the end, you have axon knob, like a doorknob. Nowadays, they don't make doorknobs like this anymore, but you know what I'm talking about. Okay, and then the nucleus in the cell body, and then the axon. So these are nervous tissue. They call them nervous tissue. On the slides, in the lab, on the lab practical exam, if I put muscle, and you write down muscular, I ask you what tissue, you write down muscular tissue, I will not give you credit. You have to specify what kind of tissue it is, a skeletal muscle. You have to be able to identify them, separate them. Anyhow, connective tissue, whatever the definition my histology professor gave me in Tennessee, whatever it is not, epithelial, nervous, what is the other one? Muscular tissue is connective tissue. That's the definition. But of course, you know the functions, you know what they are. Loose connective tissue, elastic, thin, non-elastic, cartilage. I have blood and bone. The models we have, bone and blood, different type of blood, red blood cells, white blood cells. We'll go over these later on when the time arrives. These are all blood models. Okay, so they are all connective tissue. As time goes on, you will know, make sure you study your handout, slide description, what is connective tissue in your slides. Yes, Ariana. Connective tissue. Connective CT, connective tissue. I got lazy yet. No, you're not allowed. I am allowed to get lazy. You're not allowed. No abbreviation in this class for me. No abbreviation. If anytime during the exam you write an abbreviation, I will not give you credit. Okay, bone, so on blood, and here they are. Fat cells. What is the function of fat cells? Store energy. So that's the kind of function of connective tissue. Store energy. Fight bacteria infection. What is it? Function of what tissue? Connective tissue, right? Blood, yeah. White blood cells, they fight bacteria, viruses. Ebola, you've heard of Ebola? Okay. Chondrocytes, your cartilage. Then fibroblast and elastic fiber, again in your bones, and bone, of course. All of that is connective tissue, supports you, so on and so forth. Okay, so 12 systems, of course. I will not go over them. We will, on the slides that I put up, I will ask you what system does it belong to, the microscope that you're looking at on the microscope. Okay, so couple of systems. Integumentary means what? Huh? Skin. Thank you. And then endocrine is what? The rest is simple. Endocrine is what? Your glands. Your glands. The glands of the body. Endocrine and exocrine glands. Okay, sense organs, of course, you're putting in, but the sense organs are part of the nervous system. Most textbook, but the nervous system covers like 4 or 5 chapters, so textbooks in anatomy, physiology, they break them down and put them in different chapters. Ours, they just put them all in one chapter, which is fine. That's the way we want it. We don't want to go into detail of these guys too much. But again, on your slides, make sure that I will ask you, for example, I might put this here, right? And I ask what system does it belong to? And you write down monocyte or lymphoid, monocyte is true. You write down monocyte, and then you're wrong. I ask what system, and you wrote down the name of the cell. How am I making sense? The system is immune system. I hope I'm making some sense. Answer the question during exam. Answer the question that is before you, in front of you. Let's go. Immune system. So speaking of devil, it just showed up. Okay, nervous system is the very first system we are going to discuss a little bit. So nervous system, you have a nerve bundle. You do have a slide of it over there. Make sure you look at it on the artery, vein, nerve slide. Artery, vein, nerve slide. That's the title of the slide. On the slide description, it says in there. So look at this, find this. The outside of it is connective tissue. Don't worry about what type of connective tissue. Just write down connective tissue. And that's what I wrote down in your handout for you. Okay. Then inside of it, you have myelinated neuron and unmyelinated neuron. What is myelinated neuron and unmyelinated neuron? Somebody asked me, I guess it was in this lab or the other lab, afternoon lab. So you do have an axon. We already talked about this. This is axon right here. And outside of axon, there are cells, okay, that wraps around them. Like, have you seen those rosary beads? I should bring my rosary next time. You know what I'm talking about? The string inside of the rosary, think about it. The string inside of the rosary. Y'all know, does anybody know, does not know what it is? But you all know what I'm talking about. Okay, the string inside of the rosary is axon, but the beads outside are Schwann cells. They call them Schwann cells, right there. Schwann was the name of the scientist who discovered it. Lucky him. For the rest of eternity, they're going to call this Schwann. They're changing him a little bit. Anatomy people, histology people are changing, but I don't think they will change. They will not do anyhow. But anyway, so they have nucleus, and then what happens to the Schwann cells? Both unmyelinated neuron have them, and myelinated neuron have them. They have Schwann cells. Both of them have it. What is the difference between, these are I'm answering the questions in your handout. You remember questions, exam 1? What is the difference? One of the questions, what is the difference between unmyelinated and myelinated neuron? I'm answering it right now. Okay, so unmyelinated neuron, the Schwann cell is not like a pizza dough. Think of a pizza dough. You wrap it around your arm several times, right? You can go home, grab a pizza dough, and wrap it around your arm and show it to everybody in the home. They think you've gone crazy. Say, I have a crazy zoology teacher, why not? I can do this. So wrap it around, that would be myelinated neuron. And this is your arm. Axon is your arm. Am I making some sense a little bit? On myelinated neuron, you take a pita bread, or you take a tortilla, and it just goes around how many times? Once. It goes around once, around your arm. And that's unmyelinated neuron. So what is myelin? Maybe I'll have it in the next slide or so. Myelin is a layer of fat. Cell membrane is made up of what? By what? Phospholipid. Yeah, phospholipid, 2 layers of it, right? So that a lot of phospholipid goes around axon in myelinated neuron, and unmyelinated neuron, maybe 2 layers or 4 layers of phospholipid. That's it. I hope I'm making some sense.
Myelinated has two, how many areas of phospholipids?
Many, I don't know. I haven't researched it that much, but many. That's what a Schwann cell is. So what happens, we talked about action potential, right? When you have, imagine for a second, guys, imagine both of these are myelinated. Can you guys see this? Anybody cannot see it? I have to call somebody to come up and hold this. You all can see it, right? Thank God. So imagine this is myelinated neuron, this is unmyelinated neuron. And this is the Schwann cell right here. They cut it for you to show you inside of it. This is another Schwann cell and another Schwann cell. Here is a Schwann cell, Schwann cell, Schwann cell. As I said, imagine this is unmyelinated neuron. Imagine for a second. What happens when action potential comes here to myelinated neuron, then since this is thick, action potential will jump from this node of Ranvier. Ranvier was a French scientist. It jumps from this node of Ranvier to the next node of Ranvier, to the next node of Ranvier. It jumps. That is called saltatory conduction. Saltatory, like a salsa, you dance salsa. Okay, so it jumps from one node of Ranvier to the next node of Ranvier to the next node of Ranvier. Is called saltatory conduction. So action potential gets from point A to point B, from here to here, faster because it's jumping. Am I making some sense? Unmyelinated neuron, on the other hand, it has to go through every segment of the axon. Right? You want some of my coffee? No? Tea? You want? I have tea in my office. I can boil it. We have microwave next door, and I can boil it up and give you tea, coffee. I have those Sanko instant coffees. You didn't have enough sleep last night. Get enough sleep, especially the night before exam. Make sure you get enough sleep. Okay, I don't think I'm boring, so I get offended. No, just kidding. Okay, so the action potential goes through every segment of unmyelinated neuron. This is slower. This is slower because he has to go through every segment, every pieces and bits of unmyelinated neuron. Animals like jellyfish, hydra, they have unmyelinated neuron. Animals that are advanced like us, we have both myelinated and unmyelinated neuron. So in animals like jellyfish, action potential is slower. In us, of course, we evolved and we came up with that. Yes, sir?
Why does the myelinated make it jump so much faster?
Because of the insulator. This is insulation. So that insulation causes the sodium do not get in there. Sodium only can get in here. There is a gap between this Schwann cell and that Schwann cell. Did they answer your question? It's just, that's what the Schwann cells are. It could be a quiz question, exam question, or MCAT question. What are Schwann cells? They're insulators. They insulate the axon, so the action potential jumps from one node of Ranvier to the next node, from one bit of the rosary, you have to the next, to the next. I hope I'm making some sense. Okay, let's go. Here they are. They are showing you the development of the, here is Farhad. Is that right, Farhad? Do I say it correctly?
Yeah.
Farhad. Here is many layers. You can go home and count them. Of course, it's a drawing, it's not scientific. You can count them, how many layers it is. And of course, this is a development. This is the axon cross-section of it. And here they are, neurolemocytes, the Schwann. As I said, they're changing the names because they want to get rid of the scientist's name in 200-300 years ago who covered these things, covered them and discovered them, and they named them. So instead of Schwann cells, they're going to call it neurolemocytes. Cyte at the end of the word means what? Cell. Your prefix, suffix you should know today, right? Cell. So yeah, but for my purposes in here, I probably never ever refer them in this class to neurolemocytes. You want to write it during the exam, that's fine. I'll give you credit. But I will always talk about Schwann cells. Okay, node of Ranvier. Okay, node of Ranvier. which stays almost naked axon. Okay, so here we go. We talked. Oh, neuroglia. A Schwann cell is a neuroglia. Neuroglia is a supportive cell by definition. Supportive cells. You do have them in your slides. You have astrocytes in your slides, and astrocytes are a type of neuroglia. Okay, Schwann cell is a type of neuroglia. We have many different types of neuroglia. And when I was a student, they said there is no action potential, there is no conduction in supportive cells like astrocytes and other cells. But nowadays they're finding out there is conduction. So anyhow, that's the most recent research. Here we go. Again, quickly, this is dendrites. Action potential is going down to neuromuscular junction. Here is axon knob. You do have it on the model. I showed it to you guys here. You do have slide of this. You have slide of neuron. You have slide of neuromuscular junction, where the action, the neuron releases neurotransmitter to the muscle, and muscle get contracted. Okay, I hope I'm making some sense. Here it is, answer to your question, the voltage, we talked about this. It goes, you have outside, you have a lot of sodium, and sodium gets inside of the cell. This is action potential. And right here on this chart, I hope I'm not confusing you, the resting, they call this resting potential right here. They call this resting potential right here. The neuron is at rest. It's not stimulated at minus 70 millivolt. And when sodium rushes in, then sodium gets inside. That's sodium is coming in, gets inside, and finally comes back to resting potential. A neuron cannot be stimulated all the time. If it is, Houston, we got a problem. You watched Apollo 13? It's a movie called Apollo 13. It was a lucky number, right? I played basketball in middle school. My number was 13. That's why I was always on the bench. Coach didn't put me in there. He put me one game for only half of the first half I played, and that was the end of it. I never saw the court again, except during practice. And I thought, oh, you play basketball? No, good for you. So right here, it's a heartbreaking sport, especially when they put you on the bench. It's very heartbreaking. But anyhow, so right here, if action potential keeps going, we do have problems. Parkinson's disease is one of them. Constantly being fired, a neuron, a neurotransmitter. Okay, anyway, right here, when inside of the neuron become positively charged, as I said, you put the electrode outside and electrode inside, and then the inside becomes plus 40 millivolt. This is plus, this is minus. I hope I'm making some sense. And then that's how it goes toward the end of the neuron. The end of the neuron releases neurotransmitter. And the neurotransmitter, here we go. These are the things I expect you to know. A resting state, no net flow of ions. It means right here, that's the resting state. This is the resting state. There's no ions flowing inside or outside. It is, again, I'm not talking physiology, but there is no big difference between them. It's balanced. That's what they call it, resting potential. Yes, please.
Is that diagram saying that the sodium ions are rushing in at the same time the potassium ions are rushing out?
Right. Yes. Well, I was going to talk about potassium. Did I talk about it? Yes, I did some. Yes, that's the case. Okay. But sodium gate starts to open. There are gates. What I mean, there are little gates in here, and sodium rushes in. Okay, sodium gates starts open, and some sodium enters the neuron. Many sodium enters the neuron. When many sodium, number 3, right? When many sodium enters the neuron, that's action potential.
There's so much going on, right?
I'm sorry, what?
There's so much going on, right?
There is a lot of like a positive feedback. Okay, I didn't talk about this. A lot of them, like avalanche.
And this is positive feedback because this is negative feedback, right?
No, this right here is positive feedback.
Exactly.
But your body's not taking anything to produce more. Isn't positive feedback your body you take in more? No.
Positive feedback is like avalanche.
Okay.
More and more and more of it occurs. Okay, then when you have many sodium gates open up and sodium gets in and potassium gates close, this is about the time that potassium gates close and sodium gates close and potassium gates open up. Number 4 is called refractory period, when there is no matter how strongly, right here, we call it absolute refractory period. No matter how strongly you stimulate a neuron, that neuron cannot be stimulated anymore. Okay, so that's called absolute refractory period. Okay, and the time here is about, oh, I didn't mention it, is it? None of these here. The time is about 3 to 4 milliseconds from here to here. Okay, here at the end, when action potential is coming, form the neurotransmitters. The neurotransmitters are inside, but what happens, the neurotransmitters right here, they move in a vesicle toward the cell membrane and they diffuse out, and the neurotransmitter stimulates the postsynaptic neuron. Synapse is the space between two neurons, or a neuron and a muscle, or a neuron and a gland. That's a synapse. The space between two neurons is called a synapse. So this is a presynaptic neuron when action potential initiated. This is postsynaptic neuron when action potential is going to take off and go, and this is in rest now. When presynaptic neuron delivers the neurotransmitters to postsynaptic, just like a relay. You guys watch relay on TV on Olympics. Okay, one gives the other one the baton, the next one is gone, and then this one rests. Right? Am I making some sense? Same thing. Okay, again, I'm beating the dead horse, neurotransmitter. Calcium is needed for movement of the neurotransmitters. How that is, we will not talk about it. That's not for our course. But calcium is needed. It goes inside at axon knob. Remember, axon knob, and then release and action potential is stimulated. Again, I'm beating the dead horse. All of these pictures are saying the same thing. Okay, one other thing is reflex arc that I didn't talk about it. What happens when you are stimulated in your body, you touch a hot stove, right, or something sharp, what happens? You quickly remove your hand, or when you go to doctor's office, they stimulate your knee and your knee jerks. You all know what I'm talking about.
Yeah.
The sequence of events are as follows. Okay, what happens? There is a neuron right here. It's called afferent neuron right here. It goes from the sense of the touch. It goes to spinal cord. There is another neuron in spinal cord. It's called association neuron. An association neuron sends the information into motor neuron, into the muscle, and the muscle will contract. It will jerk. Okay. So afferent neuron is same as sensory neuron. Afferent neuron, same as sensory neuron. Association neuron is found in the spinal cord, central nervous system, and takes it into motor neuron. So that would be afferent neuron would be presynaptic neuron. Association neuron would be postsynaptic neuron. And then when association neuron stimulates, just a relay. Imagine this relay team is 3. Usually the relay you watch in the Olympics is 4. But this is 3. Okay, so as afferent to association neuron, finally to motor neuron, and motor neuron becomes postsynaptic neuron, association neuron is going to rest. Okay, yes?
So is that to say that the reflex is triggered only by like the touch, like it doesn't reach your brain at all?
Right, very good. And this type of stimulus, it will not go to the brain. Then if you have burned yourself or you cut yourself, then the information goes to the brain. You look at it, the information goes, oh, you got to go put some ice on it, or you got to do something else. But initially, when the reflex occurs, the information does not go to the brain. You have different reflexes. When you cough, when you sneeze, it's a reflex. When you go to the doctor, they put the flashlight in your eye, that's another reflex. They're checking your reflexes when you go to a doctor. Yes?
Was I going to say that someone would have a reflex if they were like unconscious or dead?
The what?
Would that, would you say that reflex would exist in a body if the person was unconscious or dead?
Dead, I doubt it, but again, unconscious person, I don't know. I don't know, unconscious person could have some type of reflex. Okay, you've heard of people come back from a coma after 12 years and they claim that they saw, they knew everything. When people come to the hospital visiting them, they could not respond, but they said, yes, you came, visited to me, and you revealed your secret to me. I know your secret now. Have you heard those stories? I don't know. The wife goes to the husband, you know, I love you, dear. Please wake up. I slept with so-and-so. I did so-and-so. I cheated on you 10 times. The husband gets up. Say, okay, let's go. I don't know. Okay, I don't.
People really say
I
that?
don't. Okay. Yes, people who are coma, they
really say?
claim that they recognize it sometimes. Anyway. Okay, same thing, reflex arc. I'm not going to talk about it anymore. Sympathetic neuron and parasympathetic neuron, if you look at them, look at the list in your textbook, you will see that these are pretty much relaxing you, calming you down, and sympathetic neuron is the one that you're in fight or flight mode that we talked about it today a little bit. Okay, same thing here. Brain is, as I said, part of the central nervous system. All I expect you to know for the lab portion, or maybe for lecture portion, you don't have to know the functions of parts of the brain, but you should know frontal lobe, parietal lobe, occipital lobe, and temporal lobes. There are 4 lobes I expect you to know. There are only 4 lobes in the brain. And then on the bottom, you have cerebellum. All of this is called cerebrum. And this is on the bottom. It's called like a cow dung, layers. It's called cerebellum. And then in here, there are 2 other structures I would like you to know is pons and medulla oblongata. Medulla oblongata and pons. You have those 6 part of the brain. Farha, you had a question a long time ago.
Where's the temporal lobe at?
Frontal, right here, parietal, occipital, and 2 temporal.
On the side.
Right.